Hi, ladies, and we're back. Okay, so we left off with how many alleles control sickle cell anemia, and we said there are two, and I'm just giving that to you. There's a big A and a little a allele, and big A is what we call dominant, and little a is what we call recessive. So I'm sure that you learned many definitions for these in the past, but they are misleading, they're inaccurate, and you're going to learn my definition. It's not my definition, I didn't come up with it. But okay, so what is a dominant allele? A dominant allele is the only allele affecting the individual when the individual has two different alleles. The only allele affecting the individual when the individual has two different alleles. One more time, the only allele affecting the individual when the individual has two different alleles. Okay, and we'll get into that in the next slide, so just pause the game for a minute. Okay, recessive, let's go down to number 2. What is a recessive allele? It's the allele that seems to not affect the trait when the organism has two different alleles. Okay, so it's the allele not affecting the individual when the individual has two different alleles. So in the dominant situation, let's highlight the word affects. Affects or affecting, that would be with an A. And then in number 2, you want to highlight does not affect. Okay, so that's the difference between dominant and recessive. Dominant alleles appear to or do affect the appearance of the individual, and recessive alleles don't. And both of those situations is when the organism has two different alleles. And I'll get into that in a minute. So how many alleles control sickle cell anemia? There are 2. Okay, so let me move my little picture out of the way. So these 2 alleles, it doesn't necessarily mean that big A is on one chromosome and little a is on another chromosome. You can have big A and big A, or you can have little a and little a, or you can have little a and big A. But we have 2 alleles because we have 2 homologous chromosomes. But again, it doesn't necessarily mean that because you have 2 homologous chromosomes, you're going to have the same alleles on those 2 chromosomes. Okay. Remember, we got one of those chromosomes of chromosome 11 from your mother, your biological mother, and you got one of those chromosome 11s from your biological father. So the M and the P stand for the maternal chromosome or the paternal chromosome. Okay, so let's just give an example here. Why don't we take maybe this, I don't know, this purple band right here and this purple band right here, and we're just going to label that the person could have, say, a big A on the maternal chromosome and a little a on the paternal chromosome. Okay, so in this situation, the person has 2 different alleles for sickle cell anemia. Okay, let's go on to the next slide. Okay, so what actually makes a big A different from little a? All right, so, sorry, back up the truck. Do you remember? Do you know what makes a big A different from little a? What makes the allele different from the other one? Really, what makes the little a gene different from the big A gene? So here on the left, you have a sequence of nucleotides, and you can even take your pen and do dot, 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 because this is just a piece of the sequence of nucleotides for hemoglobin. And what we're going to focus on here is this codon right here. This codon is GAG. Okay, and so over here we can write normal hemoglobin, and you can write big A. So normal hemoglobin, big A. And this is the sequence of nucleotides if you have the normal gene for hemoglobin. And then over here, we're going to have little a. Can I do this? What do you think? Little a, big, big A, normal. This isn't going so well. Oh well, that was good fun. And then abnormal hemoglobin. So what I want you to do is take your highlighter, back up the truck, take your highlighter and highlight this A right here versus this T right here. And you can even do those highlighters in different colors because adenine is not the same nucleotide as thymine. Okay, so there's one nucleotide difference in the sequence. So this here is a piece of the gene, a piece of the gene. Okay, so there's what makes the big A allele different from the little a allele? Well, the big A allele, the big A gene, has a different sequence of nucleotides from the little a gene. So let's write that down. The sequence of nucleotides for big A or normal hemoglobin is different from the sequence of nucleotides for abnormal hemoglobin. And so you can just rewind that if you have to go back, but I got to move on because we're on a time crunch. Okay, so there's a different sequence of nucleotides for normal hemoglobin versus abnormal hemoglobin. So what does that do? Okay, so what do these two different alleles do? Again, here is your normal hemoglobin sequence. Here is your abnormal hemoglobin sequence. So you can write, again, normal hemoglobin and abnormal hemoglobin. So here's the nucleotide difference. So this codon codes using the genetic code for glutamic acid. You do not need to know that. Okay, and then here is just the side group for glutamic acid. Remember, glutamic acid is an amino acid. All of these are new amino acids, and that's what proteins are. Sequences are, sequences of amino acids linked together. Okay, and then over here, look, when you put a thymine there, you get a valine, and you can see that this is a different side group. So, different sequence could mean a different amino acid, which is going to affect the protein structure. In this case, this amino acid change is going to cause a change in the shape of hemoglobin. So again, make sure you highlight these two glutamic acid and valine in different colors. You don't have to worry about the side groups. I'm not going to say, hey, draw the side group of glutamic acid and valine. Okay, but what I do want you to notice here, and these are models of hemoglobin. So this is normal hemoglobin, and let's just model this protein as a square that has this little chunk out of it. So this is our very normal hemoglobin. This is our abnormal hemoglobin, or sickle cell hemoglobin. So it still, again, has that little piece cut out, but look, now valine sticks out. So when we did this last chapter, I didn't tell you what this thing was. It wasn't really important, but you can see that the shape of this molecule is different than the shape of this molecule. This is normal hemoglobin. This is abnormal hemoglobin. And so when you have abnormal hemoglobin or sickle cell hemoglobin, it has this little protrusion out the end of it, and look, now it can fit into the adjacent or another molecule of hemoglobin that's in the cell. This is a problem. This is what causes the hemoglobin to stick together. These hemoglobins will not stick together. Let's back up the truck. Whoa, 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 whoa. Backing up the bus just like we do in school. All right, look at this. Here's your normal hemoglobin. It doesn't have that little valine sticking out. But here, these hemoglobins have valine sticking out, and that's what causes them to stick together. Whoa, whoa, whoa. Move the bus forward. Okay, so what do the different alleles do? Those different alleles result in different shaped hemoglobins. That can be a problem. Okay, now, moving on. what effect with an E, that's a noun, what effect do the various combinations of alleles have on the organism? What does that sentence mean? Are you lost? Let's back up the truck, man. Okay, so here is our normal hemoglobin. Did I do this? Okay, here we have normal hemoglobin. So let's just say that these are the chromosomes, chromosome 11 for an organism, say a parent or whoever. It's an organism that has normal hemoglobin. There are her two chromosomes or his two chromosomes. And on one chromosome is a capital A, just like we did with our popsicle sticks. And then on the other chromosome is a big A. So if this chromosome contains the gene for normal hemoglobin, and this chromosome contains the gene for normal hemoglobin, the person is going to make normal hemoglobin. So that's what we mean when we say combinations. Combinations. What two alleles does the person have? All right, let's do another example. Suppose a person has big A on one chromosome, this chromosome, and on the other chromosome, they have little a. Well, their hemoglobin is still going to be normal because this is what recessive is. Recessive is when this allele seems to have no effect on the organism when they have two different alleles. So look, this is big A, this is little a allele. They are different. When an organism has two different alleles and there appears to be no effect on the organism, that's what we call recessive. So little a is recessive and capital A, big A is dominant. So when a person has two alleles that are the same, big A and big A, let's see if I can write this over here. They are called homozygous. Remember, homo means same. So what's the same? Their two alleles are the same. So zygos, think of like zygote. They have two alleles that are the same. Big A and little a, those are different. The word for different in Latin is hetero. So we call this person heterozygous. So we would say that since this person has two of the same alleles, they're homozygous, and because this person has two different alleles, they are heterozygous. So that's what I mean when we say the combination. What effect? The effect of big A is they have normal hemoglobin. The effect of big A, little a, they also have normal hemoglobin. Because remember, this big A is an instruction for building a normal protein. This little a is the instruction for building an abnormal protein. But there's going to be enough normal hemoglobin for the organism to function. So yes, they probably still do make some abnormal hemoglobin, but they have enough normal hemoglobin that they're not symptomatic. They don't have the symptoms of sickle cell anemia. Okay, so can I do it? Yes, I did it. I'm getting done with this now. All right, let's go to another example here. Did I not do another example? Did I forget to put that in my presentation? Okay, so again, normal hemoglobin, normal red blood cells, blood flow is totally great. Look at this lady. This person has sticky hemoglobin. So on her chromosomes, she has a little a on one chromosome and a little a on the other chromosome. So she is what we call homozygous as well. Okay, she's homozygous, but she's homozygous for the recessive allele. She has two recessive alleles, but they're both the same. And so if you have, if you're homozygous recessive, you have sickle cell disease. Okay, so here's the abnormal hemoglobin all clumped. That thread or fiber-like shape distorts your red blood cells, and then this causes clotting in your blood vessels, like in your capillaries, and blood can't flow, and that means your cells can't get oxygen, and then you've got problems. Okay, bye.